Today is April 7, 2021, and we have a special guest, Agota Horel from Hungary. She will give a seminar titled Soil Redistribution in Vineyards. Agota is an environmental engineer, and she earned her PhD from University of Alaska Fairbanks in 2009. She is currently working at the Institute of Soil Sciences and Agricultural Chemistry, Center for Agricultural Research in Hungary, and she is a senior research scientist there, and she specializes in soil science with main focus on soil water studies, soil pollution, specifically for organic contaminations, and soil water modeling in unsaturated soil matrices. She also has a research experience in soil microbial respiration and modeling studies. To remind, today's seminar will be recorded, and you can ask your questions after Agota finishes her seminar. And with that, let's give her a warm welcome, and I'm leaving the stage to you, Agota. Thank you for being here.
Thank you. Thank you so much. And it is great that I have been invited to give this presentation. Today, I'm going to talk about soil redistribution in vineyards. I really hope everybody can see the screen well. So pretty quick, anybody who didn't know or didn't look up where Hungary is located. Hungary is somewhere in middle of Europe, and the study site is right above Lake Balaton, that is in the western part of the country. This is a catchment-based study that we are doing right now. Our project is based on different parts, and I'm going to talk about only the vineyard portion, or mainly the vineyard portion for this presentation. Our study site is about 21 square kilometers. And it's a very nicely delimited area because we have only one stream. So we can actually study this small agriculturally dominated catchment very nicely. Some information is written on the left side. Elevation range is 375 through 110 meters above sea level. The Chorsa stream length is about 8 kilometers long, and it goes right into Lake Balaton, which I'm going to show a little bit later. The stream elevation is at the spring 240 meters and at the outflow 110 meters above sea levels, which is a relatively large elevation for a small stream. The precipitation is 500 through 600 millimeters per year at our meteorological station. And as I mentioned, it's an agriculturally dominant catchment. And why is it so important? Because in Hungary, this site is very famous for the vineyards and the viticulture. And we have several Hungarian types of grapes that are grown in here. I put a slide in here that what is our general information or problem statement in this area. It's mainly everywhere in Hungary getting a big issue. One is the soil erosion. At this catchment, the long-term soil erosion is one of the major issues. Increasing severe weather occurrences, mainly it's either long-term drought or a high amount of rain at a short period of time. At the low-level areas of the catchment, we have increasing occurrence of ponding of water. Also, we have, this part of the country, the grapevine plantations, and the young grapevines are generally tilled soil that is at that area. The stream outflow is going into Lake Balaton, as I mentioned. This is a recreational lake and have a very sensitive ecosystem. Therefore, all contamination, especially nutrients, coming from runoff might enter Lake Balaton, but at least the Csorsa stream. Before I go into the vineyard sites, I want to introduce a little bit more about our studies in this catchment. We have since 2015 4 land use types that we are investigating: vineyard, arable land that has crop rotation, grassland, and a mixed forest that we have at 3 different depths: soil, moisture, and temperature sensors. And in 2018 and 19, we added 2 additional land use or sampling sites: one forest that is only oak, and an apricot farm. However, we stopped since 2000, after 2020, measuring these last 2 types. We had this project that I'm talking about started in 2020, and based on this project, we added 2 additional vineyard sites to the other where we had 2015 data. And this is how the older grapevines look like. We have normally grass strips between, and it greatly can reduce erosion. However, as you can see, the younger grapevines, the tilled soil actually increases the soil erosion possibilities. However, all the plant managements are same at both sites. So actually all 3 vineyard sites that I'm going to talk a little more details in. All fertilizer additions are the same, the trimming events are same, and mostly our harvest time is done, even if it's not exactly at the same time, but with the same instruments. And some aerial pictures of the 3 slopes that we are started to investigate in 2020. One of the slope is the young plantation. The other one is the older one that you can see the grass strips. And we have the older one, I call it Furmint. That's the type of the grape that is grown in here, which we have much longer archive data because of the 2015 measurements. These blue slopes we do have at the upper portion and at the lower portion measurement points, and here too. What we have to know, the difference is it's Riesling. This one is Furmint type of grape. However, both are white grapes and very similar actually with phenologically both. These newly added long slopes are about approximately 550 meters long, while the older one is only 100, but the slope angle is much higher for this shorter study sites. It's almost 11%, while the other 2 has approximately 8% incline. So I'm going to talk about what kind of measurements we are doing. One of our main objectives is how the plant, water, and soil system is connected, how these 3 interrelations are going on. And that's what we are trying to find the questions, or just to understand it much better. One of the most important measurements are the water content. We are using water content, soil moisture, and soil temperature sensors. For these newly added slopes, it's placed at 2 depths, 15 and 14 centimeter below ground. And hopefully we are going to use it for soil moisture modeling as well. We use spectral reflectance sensors, namely NDVI sensors and PRI sensors. The NDVI, the normalized difference vegetation index, and the PRI, the photochemical reflectance index. We also use PAR sensors, which are the photosynthetically active radiation, which are we place below the canopy. The NDVI and PRI sensors are right above canopy. We were trying to make it about 2.5 meters above, somewhere around 45-50 centimeter diameter. We can actually measure continuously the reflectance from the plants. All of these sensors have, of course, the hemispherical pairs that measures the incoming radiations. We also do soil chemistry measurements. However, those are not continuous measurements. Those are only when we have event-based or combined measurements for that. We measure pH, total nitrogen, soil organic carbon, potassium, and phosphorus mainly. One very interesting part of this study is the soil microbial investigations. We have two types, one with the university, one of the Hungarian university with collaboration. We actually collect the soil samples and they use it to extract DNAs, and using this new generation sequencing, they try to do a complete genome out of these samples. And the other one is the SEER measurements, which are the substrate induced respiration, where the enzyme activities are investigated. I cannot go without mentioning the soil carbon dioxide fluxes, because this is one of the main questions that we are raised when brought the proposal of this project. And we are very interested in greenhouse gas emissions of the soil, especially carbon dioxide and nitrogen oxide, or N2O. However, our N2O measurements where we use Picarro is really, really heavy and takes quite a long time. We only can take once in a while the carbon dioxide measurements are bi-weekly during vegetation period, and once a month or once in a while during the dormant period of the plants. I also want to mention that we have done a teabag index calculations, because that's very easy and well-known method to measure leader decomposition rate and stabilization. So from here on, I want to introduce some measurement techniques that we were used, or instruments that we were used, and the results that we actually got so far from the last year measurements. I want to point out that this is just the first year of this project, and it's supposed to go through 2024. And that's going to be very interesting to see what's going to happen on the yearly base as well. We use the Decagon 5TM soil moisture and temperature probes. However, these are not anymore available, and Teros 12 is going to replace all of the injured sensors of ours. And the very first slide, I wanted to show some of our soil water content changes on all four land use types, where we actually have data since the end of 2015, because it is really important to have long-term data if we want to use modeling. Only two things I would like to point out. One is at the meadow or grassland, where it is very, very dry, even at higher precipitation events. This land use type has the lowest volumetric water content, while at the arable land, which is located at the bottom of the catchment, the water table is much higher, And at the 70 centimeter sensor is already quite often got into the saturated zone, which is either the groundwater table elevation or the capillary fringe rises. And here is the last year data for the two new slopes. One is the newer and the other one is the older Riesling sites. What we get from the water content is that the older grapevines, we get 6% higher average water content at the lower point compared to the upper point for the 15 centimeter depth and 20% less at the 40 centimeter depth. However, at the new grapevines, we got 37% less volumetric water content for the low side at 15 centimeter and 6% less at 40 centimeter depth. And why I was trying kind of highlight it is because we were expecting that at the bottom of the slope, the soil water content should be much higher than at the top of the slope, and it was not the case in our sites. And the temperature was not much different at any of these sites. At the 15 centimeter, we got all data within 10% difference, and at 40 centimeter, only 1% difference between all four sites, which is not much difference. The spectral reflectance sensors, we calculated the PRI and the NDVI as it was given in literature. We use the PAR values, the photosynthetically active radiation values, to calculate the fraction of absorbed photosynthetically active radiation, because that's actually give representative data on the plant growth and health. And here comes the two new slides, the Riesling slide, the parallel slope data, where the first graph actually shows that approximately 35% higher NDVI for the older lower points, measuring points, and 24% less NDVI for the newer lower points than the newer grapevine upper points. And again, it's kind of collaborates with the volumetric water content data that we were expecting at the newer grapevine at the lower part of the slope, different values, that higher NDVI values, and it was not the case. For the PRI values, we could say that the older grapevine is much higher than the at the lower portion than at the upper portion, and it was similar with the newer grapevine as well. The FAPAR values were much higher, corresponding with the PRI values for the older grapevine lower point than the upper point. However, the newly planted grape leaves had similar values for both measuring points at the slope. I included the Foreman sites as well, especially because of the PRI values. So the NDVI values were very similar for the upper and the lower values for both points. The PRI, however, you can see it is so much higher for the upper portion than the lower portion. And what we actually think it is because either the water content or nutrient stress for the plants. And this is one of the most interesting part of our results in this one, because it's not the first time we actually can clearly indicate plant stress by using PRI index. The PAR values were not as different. Mainly the lower values were much higher than the upper portion, which is corresponds to the older grapevine data. And this is what we were expecting after our hypothesis. Some statistics on these different slopes. All three slopes are were kind of looked together, and what we found that the NDVI values were significantly different for both Riesling sites, but not significant between the Furmint, the lower and upper sides. The PRI values were significant at all three slopes when it was compared to each other within the slope. The FAPAR values were, we can see that were not significantly different for the newer grapevines at within the slope, or the two points of the slope. We have done Pearson correlation because we did have approximately 126 days during the vegetation period when we could use all PRI, NDVI, FAPAR values, and the soil moisture and temperature values to be compared. What we found, and actually this analysis includes all 6 points, all three slopes, all 6 points that we were investigating. The water content at lower levels were positively influenced the PRI values, NDVI, and all FAPAR indices. And consequently, we are thinking that the lower portion of the water content can influence plant growth and health. The upper layer soil moisture show the strong negative correlation to the NDVI PRI values, especially in the cases of higher measurements, which were occurring at the newer upper and the old lower values. So it says also soil moisture also influenced plant growth and health. Soil temperature only at lower levels showed high correlations with the soil plant parameters. And I want to talk a little bit about the leaf area index. We measured leaf area index with a handheld septometer. It's an LP80 instrument. However, we could estimate it from the spectral reflectance values as well. We went at different times and measured all 6 points of the 3 slopes. What we found is that between sites, the difference were significant at most measuring times. However, there were no significant differences between the leaf area indices between the same slope, the upper and the lower portion. We only found a few dates when significant difference actually could be observed in these places, but overall there were no differences. And this shows with this box plot as well, but we can see that the newer grapevine were significantly different than the older Furmint points, measuring points. However, the parallel slides or the slopes were not different. I used the soil chemistry or some of the soil chemistry data as well. Unfortunately, we only had one measuring point, that was in July when we were taking. These soil chemistry data is only 6 soil samples per site measuring point, sorry. So, but we found that significant differences were observed between sites, but not between measuring points within the slope for many instances, especially in the soil organic carbon and nitrogen. But when I looked at the phosphorus and the potassium values, we found that it was within slope high differences. However, as I said, it was only one measuring point. We have actually a October measuring points that we can compare, and we did have a large precipitation event, and I don't even know, but 40 or 50 millimeter rain were falling within a relatively short period of time. And afterward, we actually went and collected samples too. And we were actually really lucky because we collected the soil samples, the second soil samples, right before the rain events. So I'm going to be really excited to see the results when laboratory work will be done on those samples. The carbon dioxide emissions. We use EGM5, which is a ppm systems, and we do have an EGM4, an older version as well, if something happens with this instrument. We do measure in rows and between rows how carbon dioxide is differ. This instrument has a built-in air temperature sensor, and we actually use these soil temperature sensors for every point that we measure carbon dioxide with. And we use this chamber, it's a closed chamber system, and we use this one to capture all carbon dioxide that is emitted from the soil. We also use a soil moisture sensor at the same time, so we can actually measure every single point, the actual soil surface, soil moisture. And these are some of our data for the last year. It is interesting that carbon dioxide always follows the temperature. As the temperature rises, the carbon dioxide fluxes rises, and as we are getting into autumn or winter, it's actually decreases. And these three additional carbon dioxide emission values are the three different slopes. We, as I mentioned, we measured these 3 points within rows and 3 points between grapevine rows, although these graphs are including just the average ones. We found significant differences between these two types of measurements. In general, we found that roots respiration were a major part of the full soil respiration values. It increased by 20% of the measured carbon dioxide fluxes. For the old grapevines, the lower measuring points showed higher carbon dioxide fluxes than the upper points, while the new plantation, it was the opposite, similar to the NDVI and the volumetric odor content data. The overall highest carbon dioxide respiration were observed in the case of the older grapevine lower portion, and the lowest for the Furmin side upper portion. And I forgot to mention, but the Furmin side is very, very eroded. So the upper version, we were expecting almost everything the lowest for the plant measurements, and some of that was actually lowest. And some of the Pearson correlation for the carbon dioxide emission. This is only data that was on this event based, and we went out and we measured that 66 points all carbon dioxide, NDVI, PRI, FOPR values, and collected the soil samples for every single measurement point. What we found is that the water content was really influencing carbon dioxide emissions. And this one was relatively believable because we had significant differences between water contents at the different sites that we were measuring. Soil temperature showed actually strong correlations, but these data are not really representative because, as I mentioned, it was only one day and soil temperature were not really in great part the difference between measurement points. So we actually could not really, I would not use this data, but the volumetric water content is actually highly influencing both the carbon dioxide and the PRI values, which we got for the entire vegetation period as well for PRI. One slide I want to show about the teabag index because we have done this one and it was a very challenging and interesting short experiment. What we found is that the lowest stabilization was observed for the Furmin site, while the highest for the Riesling lower measuring points. And it was for both the older and the newer grapevines. And for the decomposition rates, we found very similar rates for all three slopes for all 6 measuring points. And before I finish my presentation, I really wanted to show some data on the substrate industry aspiration because we have some data and my colleagues were actually doing this work, but they were using is different substrate, and these different substrates were used by the microbes and the carbon dioxide emitted, and it used different colors, and these colors were read by this microplate reader. And we only have some preliminary results, but I really wanted to show this PCA analysis of the relative catabolic response because the 3 different land uses, the meadow, the arable land, and the forest were so nicely distinguishable for this analysis. The grapevines are not as nice, and I was putting it up for this one. So I wanted to say some future work that what we are doing actually for this part of the project. We are going to use a rain simulator this year because it's relatively expensive to take this rain simulator out to the field. However, it's possible. We are going to collect soil samples and take it to the lab. It's another research institute that has this simulator, and we're going to measure the different intensities, rain intensities, how soil distribution can be measured. The NGS, the new next generation sequencing. As I mentioned, we already collected 51 samples. It was sent out for sequencing. Unfortunately, the samples were not usable, and we either have to repeat the process or we are hoping that the remaining extracted DNA can be useful and can be sequencing. We had 2 different points, measuring points for that, and we want to repeat this entire sequencing method at the end of the project. I was not talking about the hotspots, but all 3 slopes have one hotspots designated, that we are doing all the measurements or planning to do all the measurements that we are doing for the upper and lower portion of these slopes. Right now, we only did soil chemical data and carbon dioxide measurements. However, we do have all sensors, soil moisture sensors and spectral sensors and bar sensors to actually do exactly the same continuous measurements on these hotspots. And these hotspots are places that we decided, or we were looking for mainly based on digital elevation maps or chemical data that are points where soil disposition or displacement are occurring. We are doing all soil chemistry samplings. I was not talking about that, but all three vineyard slopes were like every 10 meters taken samples, and we're going to analyze it, and we're going to do this one at the end of the project. Unfortunately, the pandemic kind of put many analysis and lab work a little bit on hold. So we had laboratories closed, and it was a little bit trickier last year to do the field samplings. We also want to work with the our different departments. They have a UAV that can be used for area surveillance, and we really hope that with a collaboration project can surveillance the entire, I would say the entire catchment, but I would be really happy with those land uses that we are actually using for measurements. And one day I really hope for a PhD student or a master students that are going to use satellite NDVI data, and we compare our near field measurements, work some magic with that. So these are mainly the soil related studies in this catchment and in this project. I wanted to write a few people to thank. They were helping me either with lab work, field work, or providing me with information for this presentation. So thank you so much for your attention, and if you like, I would be ready for questions.
Thank you so much, Agota. That was a great seminar. I enjoyed and learned a lot from your seminar.
Thank you.
What questions do we have for Agota today? Yes, Marshall.
Hi, Dr. Agota. I appreciate your talk. I really enjoyed it. I would like to ask you about the community level physiological profile, the microREST work you did. We're using that in my lab, and I haven't had a student use it in a while, but I've worked with it before. I was wondering if you would speculate. I don't know if you said what, you had two different symbols in that figure. Yeah, upper and lower. Was that different soil depths? I don't know if you had explained that or not.
No, the upper and lower means that we use different slopes. So the top portion, upper portion of the slope, we were collecting several samples, and the lower portion of the slopes. So, however, this is not what I have done. This is my colleague's PhD student done it for us, and this is still kind of preliminary. But if you have any questions, please send it to me, and I can ask. So I can answer any microbial related questions from people who are working with microbiology.
Yeah, it's really fascinating to see, like you said, how separated those land uses are based on how the microbes are using the different resources you fed them. I've never seen PCAs quite separated as much as those are. It's really neat. I'll send you some more follow-up questions.
Please do. Please do, because I'm almost as curious as you are. I really want to know the answer.
I'm going to jump in. I have a question that came through the chat box from Dr. Manu. Have you established any relationships between some of these soils and environmental parameters and grape quality?
These are actually right now relatively preliminary studies. So what I was using for these correlations, I only did the Pearson analysis. So we did find that, for example, for carbon dioxide emission, both the temperature and the water content can greatly influence. We are hoping to have more data on the soil chemistry, because what I showed, that's only one measurement time. So I really hope that our database is going to grow by, I would say, by the end of the year, but that was last year that I was hoping for. So I really hope mid-summer we can catch up with many of these analyses, and our statistical data will be more reliable as well.
There's also a question from Ala Kalal. She thanks, and she asks if you measured enzymes activity. If so, which enzymes activity did you measure and what were the findings?
Yes, there are some measurements of the enzyme activities, but I have not done that one personally. So the same girl is doing actually who did this PCA analysis, and they're going to tell me the answer when it's going to be available. I really hope soon, because we have done all the measurements. My colleagues were helping them preparing everything to fasten a little bit these measurements, but I don't have any additional data as of yet. I really hope I'm going to have, because all of these soil samples were taken at July, which is one of the most active part of these vineyard sites in Hungary. And we took one after harvest in October to actually have some comparison with this within time as well. We also planning to repeat these measurements 2 years from now. Maybe the third year of the project, if something happens at the fourth year, we can still do any repetition.
Any
Sorry.
other? Yeah, go ahead. Sorry.
No, it's okay. That's what I wanted to say. That sorry, I cannot provide an answer as of yet.
Yeah. Okay. Dr. Menu has a question. Andrew Menu, what type of studies will you do with unmanned aerial vehicles?
That's something that we are planning right now. So we have to sit down. What we really want to do is to get the full spectra for above these grapes. So we really want to know that how our continuous measurement points, especially the NDVI and PRI values, can relate and how heterogeneous the entire area. It's on a larger scale. So I'm not quite sure what these drones can do, but I've heard that they have some brand new instrument attached to it, something about the infrared, and we hope to enhance the measurements to soil water content as well, because we do have people who are working with that. But that's not going to be my responsibility. I'm just going to provide the research side and the additional data that they might need.
I see.
So it's actually this project is pretty multi-collaborated projects with microbiologists and different departments from other institutes as well.
Okay, any other questions?
Dr. Horel, I shared your surprise about the difference in soil moisture in that one field between the upper and lower. I'm trying to think about kind of the parent material from the pH. Am I guessing right that it's deep limestone residuum for that whole area?
Yes, yes, that one is. There is the pH. Yes, actually the pH were much higher, but I have not checked the statistics on that. I'm guessing it's going to be statistically different than the other two, but if you can see that the lower and upper portion, the pH is very similar for the same slope. So I don't think that's going to be the reason. I'm actually hypothesizing that maybe the soil water might not be able to stop at the bottom of the slope, so like the other ones, and the runoff might go further down, and maybe that's the reason that the lower portion will not show as much difference. But it still doesn't explain that why is it so high at the upper portion. So I think it's going to be something with the slope angle at the different parts that I'm going to watch, but it's not proven yet. So we actually have to do either longer term experiment or more deep in-depth analysis what we have right now.
Right. But pretty consistent clayey texture.
Yes.
Yeah.
Yes, clay. Yes. But some of the soil physical parameters are not measured yet. It's already in the lab and is waiting for to be measured. So I haven't even, or I couldn't even put up data on how the soil grain, soil distribution is in there. So, and I really hope to measure intact soil samples as well, because we are hoping to do, or I'm actually hoping to do some soil moisture modeling. And for that one, I'm going to need quite a few additional data on soil physical properties that I don't have right now.
Dr. Horel, I also had a question. What was your take on for the variation in phosphorus? What were the major factors affecting the variation?
At the Furmint, the largest difference was at the Furmint site, and one of my take is because we do have archive data on this particular slope, that the upper portion is so degraded. It's very stony, while the bottom portion is really, so the slope is coming really, really inclining, and the bottom is kind of leveling out a little bit. So I think soil distribution is occurring much higher rate at this slope than the other slopes. And when fertilizer addition is occurring with mainly organic manure, then after rain events, it's actually going to accumulate at the lower portion of the slope. So, and our archive data is actually trying to prove this.
Thank you. Thank you for your answer. Other questions for Dr. Horel? Okay. Dr. Horel, thank you very much for attending and joining us today. It's been such a pleasure and I enjoyed a lot. Before we finalize the meeting, I would like to remind that we're going to be having Dr. Lee Brass as a speaker, and I'll look forward to meeting with everybody again. And thank you very much for attending today. Thank you. Thank you again, Agota.
Thank you so much for inviting me.
Thank you.
Have a great afternoon, everybody.
Enjoy your night. Sleep well.
I will. Thank you.
Szép álmokat.
Bye. Szép álmokat. Köszönöm szépen.
See ya.